Hello everyone, my name is Devin Johnson. Today I'll be discussing my presentation on understanding how tissue stiffness drives fibroblast activation in disease. Just a quick introduction, I focus on fibrosis. So what is fibrosis? Well, it's a disease in which an organ experiences either an acute injury or inflammation. This then causes the development of scar tissue, which is built off fibroblasts, and this increase in stiffness then causes a decrease in functionality of that organ. So as I mentioned earlier, when you're comparing a healthy tissue to a fibrotic tissue, something you'll notice is the difference in presence of fibroblasts. So typically in healthy tissue, these cells remain dormant. However, in a diseased environment, they become activated due to differences in the ECM, which then causes them to form this scar tissue that's stiffening the organs. So my project looks to better understand what's driving these cells to progress through disease and is there a way to prevent it. Now, the specific aims of my project are to first find a biomaterial that we can confirm is paleoelastic as well as find ideal stiffnesses. We then want to run a mechanosensitive trial with these fibroblasts to confirm that they are in fact mechanosensitive. And then lastly, we want to confirm the effectiveness of this platform with a drug screening trial. This is the experimental timeline. We'll first discuss the hydrogel portion. Now, for the hydrogel, we had different stiffness hydrogels that we did an angular frequency sweep on. This then gave us the storage modulus, and based off its linear behavior, we could assume pure elasticity. We then multiplied the average of the storage modulus for each gel by 3. This then gave us the Young's modulus, which is the stiffness the cells experience. The two gels we used were a stiffness of 8 kilopascal and 24 kilopascal. We then seeded these fibroblasts on these hydrogels and stained for collagen 1A and alpha SMA, as these are two hallmark stress proteins that these cells express. We also gave them a control vehicle in TGF-beta media. TGF-beta is transforming growth factor beta, which is a stress protein that initiates this activation response for these fibroblasts. And the key takeaway from this is that on a stiff environment, they're increasing the expression of these proteins, but synergistically with TGF-beta, we see an exponential increase in the amount of expression. We then confirmed this by running a PCR to look at the ACTA2 and COL1A1 genes to see that they were in fact upregulated on stiffer environments as well as with the presence of TGF-beta. Lastly, we want to run a drug screening platform to confirm this platform's effectiveness, and we used 3 inhibitors to do that. The first was verteporfin, which prevents aptase from shuttling to the cell nucleus. The second was Y7632, a ROCK inhibitor, which prevents the ROCK enzyme from allowing this actin myosin contractility to occur. And lastly was the trunculin A, which prevents these G-actin monomers from forming this F-actin filaments. This was the experimental timeline, and they were exposed to these inhibitors for 3 days. As you can see from the data with verteporfin and ROCK inhibitor, the mechanosensitivity of these cells was removed, telling us that they do play a critical role in cell activation. However, with the trunculin A, we see that there's still mechanosensitivity present, telling us that the actin polymerization may play less critical roles we had thought. In terms of future directions, this will be used as a baseline for 3D cell culture and patient-specific treatments for drug screen and toxicity tests. Thank you.